So this week's lesson is mutation. So let's have a look at our definition first of all. A lot of people really simplify this and have like a eukaryotic bias. So I'll put here, it's an alteration to the nucleic acid sequence of the genome of an organism. So nucleic acid, this is generally DNA. Yeah, if you're a eukaryote, however, it can also be RNA. Remember, that's where we've learned about HIV. So viruses mutate. The coronavirus that causes COVID, that mutates, and that's an RNA virus as well. So it's important to know that it's not just DNA; it is RNA as well. It's also changed the amount of DNA in a cell. This is where we would learn about. Remember, in year 12, you did about non-disjunctions. So yeah, this is a non-disjunction. There are more that we're going to meet in year 13, but it seems we're doing this before the mock exam. Let's just focus on year 12 stuff. So yeah, so non-disjunction mutations, and we'll tackle them at the end. So then we get to causes of mutation. The error here is people say that, well, it's like I'm damaging the DNA, and therefore we need to give a reason as to why the DNA gets damaged. And people talk about radiation. So they'll say radiation can cause damage to the DNA. However, we need something before radiation. Yeah, so that might be the word ionizing. In biology, I just call it high energy radiation. High energy radiation. And a really good example that's really significant to biology, because we're exposed to it all the time, is UV. Yeah, ultraviolet radiation, UV light. And that's really significant to, in particular plants, because they hold their gametes, their sex cells, in the sunlight, as in the part of a flower, aren't they? Part of a flower structure, and therefore there's a risk of mutation. So yeah, so we always talk about high energy radiation as a cause. However, that's only our first bullet point up here, an alteration to the nucleic acid sequence. So for example, UV light causes adjacent thymines in DNA to react with each other. It causes a kink in the DNA that needs to be fixed. And when the DNA is fixed, it can cause, the wrong nucleotide can slot in. So it's knowing that as well. So we've got high energy radiation. It can just also be a random copying error. So yeah, occasionally you'll get the wrong nucleotide slotted in when you're copying the DNA. And therefore, if you're having to do lots of copying of the DNA because there's lots of DNA damage being picked up, then there's a higher chance of getting a copying error. And lastly, also things can go wrong in meiosis. And that's our second bullet point up here, because that can cause a non-disjunction mutation. So yeah, this is a meiosis error. But that would change the amount of DNA that's in a sex cell, a gamete, but it's still a mutation. So that's the cause of mutations. Then it's types of small-scale mutations. So I'm breaking these mutations down into small and large scale. The meiosis one, where we're going to change the amount of DNA in a cell, is going to be a large-scale mutation. So we're going to focus on these small-scale mutations. And in year 12, there's two you need to know about. There's the deletion and the substitution. However, the addition is just the same as a deletion, as in it has the same effect. So deletion, this is where we're going to add, not add for a deletion, is it? Sorry. Yeah, so we're going to, remove a nucleotide. Often it's called base as well. It doesn't actually matter if you say base or nucleotide. Normally have to be really careful with nucleotide and base, but here, as I could be talking about the sequence of bases and therefore the base has been removed. But technically you'd be removing a nucleotide. You don't just leave it with if I've got a segment of DNA, so here's one strand, it's A, T, C, well, that looks more like a G. let's make it a G, 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 C, A. If I deleted one of these, then I wouldn't just leave it like, let's get me a deleted pen. There you go. That's too big. Let me delete pen smaller. There you go. So if I deleted the guanine base, it doesn't, it doesn't leave a gap there. In effect, you would remove the entire nucleotide and then join those two ends together. Yeah, so it just misses one out. Again, not really significant for anything that we're going to meet, but just in case. And substitution, this is where we're just going to change one. Yeah, we're going to swap one nucleotide for another. So I always imagine that I might have, a parent strand of DNA, and it's got A, T, C, C, and then I'm building a new strand, so a G comes in, a G comes in, but occasionally you might just get the wrong base coming in, and I get another G there instead. So I've got this kind of non-matchingness, and that might sneak through, and then you've ended up with a different base here, or you've had a mutation that's damaged the DNA really, really badly, and therefore, again, we're going to look at scenarios later. I might have just lost that bit, and I don't know what the parent strand was, and therefore, it might have a guess. Right, I'm going to put an A there, and then before I know it, I've got another base has been substituted. So there's numerous causes of it. Just react to whatever they ask in the question for them. So we're going to meet how these types of mutations impact on some scenarios on the other side of the sheet in a bit. So we're just going to relate this to the genetic code as well. So remember in the syllabus it talks about the DNA code is described as degenerate, non-overlapping and universal. So two of these are really relevant to mutation. Degenerate is really significant because this is where multiple codons, codon is in a minute, code for the same amino acid. A codon, this is 3 coding bases in mRNA. Remember, mRNA has come from the DNA, so it's, I could say that the DNA triplet codes for the same amino acid as well. So sometimes we can change one of the bases in the DNA. That will change one of the bases in a codon of the mRNA, but it will have no effect on the amino acid it codes for because multiple codons will code for the same amino acid. And that's because we have 4 bases. We use them in 3s. Therefore, there's 64 possible codons. And there's 20 amino acids. So therefore, we have this overspill. So there's numerous ways of coding for certain amino acids. Next one is non-overlapping. This one's important as well in mutation. This is that the ribosome always moves 3 bases or a codon. So ribosome moves, stroke advances is probably a better word, 1 codon each time. So it moves from codon to codon to codon to codon. It doesn't advance 1 base. Therefore, again, that can have implications for basically causing a frame shift. So if you deleted 1 base, then you're out of your reading in 3 sync. Okay, if you remove 2 bases, you're out of your reading in 3 sync as well. If you remove 3 consecutive ones, then you've just removed a codon, so it wouldn't matter. So often it's all about removing one base, and therefore the ribosome still reads in 3s. It doesn't know where a codon is. So if I had a sequence of, here's a bit of mRNA. So let's give it an mRNA code. So I'm putting no T's in it now. So yes, the ribosome will bind on, and it will just always, again, it always starts at AUG. So it's going to start here, and then it's going to advance 3, 3, 3, 3. If I got rid of that A here, and we joined it back together again, so let's assume that there's no gap there, it's now going to read it UUA, it's going to read that 3, followed by that 3, followed by that 3. So it's reading out of sync, and that's called a frame shift. And that would lead to a vastly different protein being made that's going to be completely non-functional. Universal doesn't really, has got nothing to do really with mutation, because it just means that all life on Earth uses the same codon system. So all life uses the same codon system. Same codon system. And it's all life except some very rare bacteria. This is really important when we get into genetic engineering in year 13, where I can move a gene from one species into another and it still makes the same protein. So let's look at some examples, because this is where the interesting bits are, I think. So let's do our first one. So we've got a single base deletion with an intron of a gene. Well, introns, these are non-coding. Yeah, they are removed in splicing. So it's removed during splicing. So it never reaches a ribosome. So it's going to have no effect. Yeah, so when we do splicing, we remove introns, we join the exons together, and therefore there's no effect. So yeah, this has no effect on protein. Yeah, as introns don't reach the ribosome. A lot of people think it'll cause a frame shift, but yeah, it never reaches a ribosome, and the ribosome is just going to attach the first coding codon, and then it starts moving across the mRNA. So it's just reading introns. It's just reading exons. The introns never get there. So it's completely irrelevant to the protein. It's not irrelevant to us because it means that we've got genetic differences, and you can therefore measure that as a difference. Next, we've got a single base deletion. All right, so I'm thinking frame shift again, in a non-coding section between genes. Right, so this is going to have no effect as well. So here's some DNA. Here's a gene. Let's call this gene B, and let's put gene A up here. So there's two different genes, and we're going to do a mutation between these two genes in a non-coding section. So we're going to do a mutation here, and it's a deletion mutation. So surely that's going to cause a frame shift. So every gene further down here is going to be affected, but that's not the case because these bits between genes are never read. If I want to read this gene or transcribe this gene, we need the enzyme RNA polymerase. So RNA polymerase, let's give it its promoter region. So this is its promoter. It's like the on switch for a gene. Well, RNA polymerase will come and bind to the promoter. So this is RNA polymerase. And it will transcribe the gene. So it will move towards the end of the gene when it hits the end, called a terminator. So all RNA polymerase does is it moves across the gene and then off again. So it comes on and off. So it only reads between the genes. So this will have no effect. Yeah, so it's this mutation, deletion in a non-coding section between genes has no effect on any protein, as these parts are never read. Parts are never transcribed. And if you think it through, it just wouldn't make sense for, if RNA polymerase kind of just went on the start of a chromosome and went all the way along all the time, then every gene would have to be expressed. And when we don't do that, most of the genes are turned off. as in, if you're a muscle cell and gene A is the amylase gene, that gene will be wrapped tightly around histones and not expressed. So we kind of turn genes on and off by wrapping them around histones. And in this case, a mutation here would, every single gene further down would then be just a nonsense. It just mean that the cell would die. It wouldn't be viable. So we only transcribe genes with available promoters that RNA polymerase can bind on and make mRNA from. Remember, splicing comes next. So that's a really good one. So those first two haven't resulted in a change to the protein, and 3 might not do as well. So we've got a single base substitution within an exon of a gene. So this may have no effect. If the change codon the same amino acid calls for the same amino acid, putting AA for amino acid, and this is due to the degenerate nature of the code. Okay, or it may cause one different amino acid in the protein. Depends if the change has resulted in the same amino acid or not. So that one depends as well. Number 4, I've only got 3 more to go. A protein made up of 200 amino acids has a middle section where 50 amino acids are out of sequence. This was due to 2 single point mutations. Explain how. So how do I get 50 different amino acids? So let's draw it out again. So there's my mRNA. So here's my mRNA. And therefore, the middle section, so I've got like a section between here and here that's different. Yeah, so it's how on earth do I get this? Well, if I did a deletion here, followed by an addition, I'd have a frame shift in between. So this is where I get a frame shift. So I've got normal, then I get a frame shift for 150 bases that would call for 50 amino acids, and then I get back to normal again. So this is where it's worth knowing that's our deletion, that's our addition. If one's followed by the other, you cause a frame shift and then you restore the frame shift. If this was 3 single point mutations, then you'd have a deletion followed by a deletion followed by a deletion, and then you get a frame shift to the next deletion. You would continue the frame shift because you're still out of 3s, but once you've done the 1, 2, 3 deletions, then you're back to normal again. But I can't do it with 2, so it must be deletion, addition, or addition, deletion. It wouldn't really matter which way around they were. However, on Year 12 syllabus, addition's not mentioned, so they couldn't ask an addition version on a Year 12 exam. However, they could ask the 3 single point mutation ones instead. So that would, as I put there, a deletion followed by an addition that are spaced out by 150 nucleotides, and that'd be on a mark scheme. Knowing 50 amino acids, and the question would say explain with reference to the mRNA. So you need to know that that 3 bases code for, not are, code for an amino acid or 1 amino acid. Often though. So if this is a 200 amino acid protein, it's made of 600 bases in the mRNA. The DNA, the gene, might be longer because of the introns have been removed. That's number 5. A single point mutation resulted in an allele of a gene that codes for an enzyme responsible for making the fur pigment melanin. That should be non-functional, sorry. It should be to be non-functional. Change it on the sheet before I print it off for you, sorry. Explain why the cat is not an albino. So I've got a single point mutation. I've got a gene now that codes for a non-functional enzyme. So I've got an enzyme. So it'd look something like this, where I've got a white pigment that gets converted into a brown pigment called melanin. And to do that, I need an enzyme. So we're saying that this enzyme is not functional. So we're going to make this enzyme non-functional. So the active site doesn't fit the white pigment to change it into a brown pigment. So surely the animal, the cat, should be an albino. It should have white pigment in its fur, not brown pigment. However, we have genes in pairs, don't we? We have two alleles of each gene, assume it's not on a sex chromosome. So so there's another enzyme made from the other homologous copy of that chromosome, and therefore there is a functional enzyme, so it will code for the brown pigment. Most alleles of genes are in pairs. So there is still a functional enzyme from the other gene. The other, I should say allele of the gene, allele of this gene. I basically created a recessive allele. So this individual is now a heterozygote. So it's got a dominant and recessive, it's got a dominant, and it's got a recessive. Dominant will be the normal enzyme, the recessive will be the non-functional enzyme, as in the gene that codes for them. However, if you have a dominant allele, you have the characteristic. So that's another one where you think, you can forget the bigger picture. And then lastly, because I'm getting on for time, sorry, how can a gamete be produced with an extra copy of chromosome 21? Well, this is our non-disjunction. Non-disjunction occurs during meiosis. It said gamete in the question, so right, I'm bringing in meiosis. This is where spindle fail to separate. Oops, what are we doing here? Separate I think we're through now, centromeres. Meres of chromosome 21. So the gamete produced has two copies. So basically I've got a cell, I've got chromosome 21s there, I've got my centrioles, there's my spindle, it's attached to that side, it's not fully attached to the other side, and therefore both of these get dragged to the south pole, and then they separate later on, and therefore I've got a gamete with two copies, and therefore if it then goes on to fertilise, will be fertilised by another one with one copy 21. We've got trisomy of chromosome 21, and that's all how Down syndrome works. So I'll stop there because we've gone on for plenty long enough there, almost 25 minutes. But hopefully it's something useful. Take care.